Hello, good afternoon. My name is Valeria Malta and I'm with Naomi Rivera to present our degree project. Nanoencapsulation of ampicillin and doxycycline with chitosan as an alternative for administration upon bacterial infections.
Many of the drugs currently on the market present multiple disadvantages in the form and in the route of administration. Because of this, it's increasingly difficult for people to accept it or to complete it. At the same time, other problems will continue to arise as a result of this disadvantage. An example of this is bacterial resistance. Bacteria creates resistance to a drug or the usual dose of drugs. So the next time you have an illness from the same bacteria, it will require even more doses or a stronger drug. There are many reasons why people don't complete their treatments, their antibiotic treatments. And our project aims to offer an alternative to the ways and the routes of administration.
In a Cuban magazine of military medicine, cases were reported of soldiers who died after having created bacterial resistance due to lack of medication or bad administration. In a study carried out by the Latin America Pharmacy magazine in 2010, it was published for the brand of Zoology. It has been possible to manufacture chitosan films to release tetracycline and help treat eye infection to river fish.
Where is our project applicable? On the work site, people travel, and in these trips, people forget to take the treatment or they just don't contemplate it. Older adults have the same problem, as most begin to suffer dementia and either forget to take the treatment, or it's just difficult for them to swallow tablets. Children don't accept some routes of administration, such as parenteral and oral route, and as most of them suffer from stomach infections with vomiting, the oral route is not at all effective. In general, painful and uncomfortable routes of administration are not accepted. And as soon as the symptoms are dissipated, the treatment is terminated.
Chitosan it is a substance that contains high potential for charge and transport molecules. In this same way, the molecules can have a good stability, absorption, biocompatibility, and low toxicity. We decided to use ampicillin because it is the most widely antibiotic in Mexican market, and doxycycline because in the history we discovered that has a good affinity with chitosan. It is important to emphasize that chitosan has an antimicrobial, antioxidant, healing, and unhealthy characteristics.
The development of nanoencapsulations of some of the most widely used antibiotics will expand the options for their administration. Nanoencapsulations present multiple advantages that make them excellent candidates and good options to be used in other possible new routes of administration. In addition to their characteristics, their absorption and proper functioning in the organism will not be in doubt.
For the elaboration of nanoencapsulation, we will use the unihilation method. This technique relies on unique interactions between positive amino groups of chitosan and negative groups of the molecules. Sodium tripolyphosphate works as an ionic bridge to bind chitosan molecules and thus coat the antibiotic and encapsulate it.
Methodology of the project. 6 solutions of acetic acid are prepared at different concentrations, in which 6 solutions of chitosan are also dissolved at different concentrations. These solutions will be resting for 24 hours, filtered, and their pH will be adjusted. Then, the antibiotic is prepared in 3 different concentrations. We will have 12 solutions, 6 for each antibiotic. The tripolyphosphate is prepared and will be added to the solutions of chitosan and antibiotic while they are in constant agitation, at different revolutions per minute. The final solution will be centrifuged.
To this nanoencapsulation, we will make a UV visible spectrophotometry test to know the concentration of antibiotic that they have inside. We will also microscopically monitor each different concentration of chitosan to see the evolution of particular site of the nanoencapsulation.
Our hypothesis is to test the effectiveness of the nanoencapsulations, and that their performance is between 50 and 65% antibiotic encapsulation.
In this research, we have learned that bacterial infection is one of the most frequent diseases in Mexico. So we decided to focus and give another form of administration, more comfortable and safe.
In conclusion, the expected results are that most of the antibiotic is encapsulated with the chitosan, and its encapsulation satisfies the characteristics of a nanoencapsulation in order to be able to verify its effectiveness. Thank you for your attention, and we hope that this presentation and this project has been to your liking.